do we, let's see, I apologize for having to fumble here through this, but Madison, Madison, come closer. Ladies and gentlemen, Madison Brown, a 2023 Doyle Students Recipient.
Hi everyone, my name is Madison Brown. I'm studying at BYU in the biology department with Dr. Rachel Wood. I'm here today because I won the Doyle W. Stevens Award last year, and I will be presenting on the progress of my research, which examines the effects of invasive plants on wetland microbial systems, as well as the effects of invasive plant removal. Okay, so a little introduction to my project. Wetlands are extremely productive and important ecosystems, not just for the environment, but also for us. They act like large sponges or kidneys for waterways, storing excess nutrients and pollutants. Great Salt Lake wetlands are an especially important stop for millions of migrating birds, which we learned a lot about today. Wetlands are also powerful players in greenhouse gas flux. When properly taken care of, they can provide carbon sequestration and climate mitigation. But when they are disturbed or drained, they can become massive sources of greenhouse gas production. This is a picture of Bear River Migratory Bird Refuge. And wetlands are faced with a lot of disturbances globally and right here in Utah. Many have been drained for agriculture and urban development, and many that are left receive the harmful runoff from such development. Then, of course, there's the familiar water diversion issue, keeping wetlands from receiving the water that they need to naturally maintain themselves. Last on my list is the invasive species like Phragmites australis, which can entirely change the dynamic of a wetland system, wreaking havoc on ecosystem processes. The map on the bottom shows the spread of invasive Phragmites in the United States as of 2015. As you can see, it's quite pervasive. So why do I study the soil microbes in wetlands? The soil and its microorganisms are the base of the ecosystem. They are the drivers of important biogeochemical reactions, some of which can only happen in the absence of oxygen, fostered by flooded conditions in wetlands. I look at the nitrogen cycle in particular because it is almost completely driven by microorganisms. And excess nitrogen is an environmental issue causing eutrophication in aquatic systems. Anaerobic microbes perform denitrification in anoxic environments like wetlands, returning excess nitrogen back into the atmosphere. The soil community is connected to the vegetation present in a dynamic relationship. Though often overlooked, the soil microbial population has a large influence over plant growth and success. And in turn, the plant species present can influence the microbial structure in a number of ways. For example, when non-native plants invade a system and reduce the diversity of plants present, it is seen that the microbial diversity also declines. Which brings me to the invasive plant, Phragmites australis subspecies australis, that tall, fluffy plant that grows along the highways and lakes and blocks the views. I think we all know it a little too well here. Introduced from Europe some time ago, this common reed established itself here in Utah after a series of floods in the 1980s devastated the native vegetation. Phragmites grows tall and thick, as you can see in this picture here, making it difficult to access waterways for recreation. Since control began in about 2006, at least 55,000 acres of Phragmites have been removed, and that number might be different. I'm not an expert on this, but I know it's at least 55,000 acres of Phragmites have been removed. But as with most invasive species, Phragmites is very difficult to manage, and so remains a common sight. Years of ignoring Phragmites in the early stages of its presence here in Utah led to this powerful competitor becoming a huge problem in many of our local systems. This fast-growing opportunist crowds out and shades out the native plants that local fauna rely on, leaving poor quality food and shelter. Because the areas surrounding the Great Salt Lake are mainly desert lands, the wetlands are especially critical habitat for approximately 200 species of water birds, and Phragmites has made a large dent in the available habitat. Additionally, the deep and extensive root structure of Phragmites changes the soil structure, and its rapid growth likely impacts nutrient cycling and soil nutrient ratios. Phragmites has also been accused of drying out wetlands and small streams, further reducing water flow to our water-starved lakes. I don't know how well you can see these graphs here, but from our preliminary data, there may be some merit to these claims against Phragmites. When we categorize sites by the simple criterion of presence or absence of Phragmites, we can see that sites with Phragmites are associated with noticeably lower water levels and more oxygenated soils. That's what the ORP is on the right, a sign that they are drier. There's usually an observable oxygenated layer of soil surrounding the roots of aquatic macrophytes like Phragmites. And Phragmites has a dense root structure which could account for this, as well as high relative rates of transpiration compared to native vegetation. But we also have to remember that this is not evidence of causation here. We cannot tell from this data whether Phragmites dries out soils or whether it simply prefers drier soils than the native vegetation. While invasive Phragmites has certainly done a lot of damage in our aquatic systems, it can provide some services itself, surprisingly. Because it grows so quickly, it can sequester nutrients and carbon and heavy metals better than many of the native plants. Pictured above is the thick root system, which stabilizes soil to help prevent erosion and soil loss. A good property right now in the coastal wetlands of Louisiana, where they are experiencing rising sea levels. While Phragmites reduces habitat for many native fauna, there are some animals like birds and snakes that can use Phragmites as habitat. And Phragmites doesn't seem to mind the disturbances that threaten many of our wetlands and native vegetation. It seems to thrive on disturbance, persisting under conditions that are inhospitable to more sensitive plants. For all of these reasons, Phragmites is favored around the world in constructed wetlands for the treatment of wastewater. Let me now give you a short introduction to the methods used to remove Phragmites on Utah Lake and the Great Salt Lake. And I'll keep it really short because I know Keith and Karin will do a much better job right after my talk. The most common method is the application of herbicides, followed by the mechanical removal via trampling and mowing. Also commonly used are cattle grazing, controlled burning, and seeding with native vegetation after these other methods. These treatments are widely used on Utah's wetlands and have been instrumental in successful Phragmites removal. But are there unintended consequences to these treatments that might result in trade-offs to Phragmites management? Bear in mind that I am not an expert in this. And there are a lot of people in this room who have way more experience in management than, well, I don't have any experience in management, but there are a lot of people who know far more about this than I do. But as far as I'm aware, removal treatments are expensive and time-intensive, and cannot possibly be applied everywhere Phragmites exists right now. Some of these treatment methods are non-selective and can accidentally be applied to the wrong sites, killing native plants and inviting Phragmites to invade. Successful removal requires multi-year treatments, and without consistent effort, Phragmites quickly grows back. Like I said before, Phragmites control is tricky, it's complicated. Our lab is interested to see if such treatments might have some direct and indirect effects on soil microbial activity and nutrient cycling within the ecosystem. It's important to get Phragmites under control before it causes irreparable damage, but it's also important to consider how Phragmites treatments might affect microbial community structure and associated ecosystem services, such as healthy nutrient cycling and carbon sequestration. Microbial communities are traditionally overlooked in invasive plant management, and such an important part of the ecosystem should be studied in depth in the presence of ecosystem shifts. You might be wondering why changes to the soil microbial community would even matter. The soil community provides important ecosystem services like decomposition and nitrogen fixation that make nutrients available for plant use, and they can ameliorate high nutrients via processes like denitrification. But they can also be involved in the production of harmful toxins like methylmercury, a major concern here in the Great Salt Lake. They are also involved in the production of greenhouse gases like carbon dioxide, nitrous oxide, and methane. Soil microorganisms naturally produce these gases under a variety of conditions, but imbalances and sudden shifts in the ecosystem such as a permanent wetland drying out, for instance, can lead to sudden and massive releases of greenhouse gases, further exacerbating climate change. And as is commonly seen in ecology, there are beneficial microbes in the soil that are usually specialists, sensitive to changing conditions, and the generalists that proliferate under challenging conditions are often much less beneficial for soil and plant health. Which leads us to my research questions. How do the microbial communities differ in structure and function between native and invasive plant dominance? And how might invasive plant removal be affecting the microbial population and its functional capacity, especially in relation to nutrient cycling? This two-pronged approach seeks to identify the changes to the soil and its communities after Phragmites invasion in a range of systems and conditions, and any changes that might occur after the plant is removed. Both of these ecosystem changes have the potential to interfere with nutrient balances, and it's important to gather data to explore such potential effects. This is a simple visual representation of how Phragmites might affect the nitrogen cycle simply by producing more biomass that then dies and accumulates in wetland. To answer my questions, I need to compare microbial diversity and nitrogen cycling between sites with native vegetation, sites with untreated Phragmites, and sites that have received treatment to remove Phragmites. So, in 2022, Rachel Wood and the Abbott Lab conducted a massive sampling project, collecting soil and water from 75 wetland sites around Utah Lake and the Great Salt Lake 3 times in one summer. Sites were selected with the help of Hambrecht and the Division of Wildlife Resources. Our sample sites fall into the 3 categories mentioned previously: reference sites with native vegetation, and Phragmites-dominated sites that have not been treated, as well as Phragmites-dominated sites that have received treatment to remove Phragmites. These sites cover a range of salinity and land use, including both agricultural and urban land use in a contributing watershed. With such a wide range of environmental variables, it's likely that we will be able to identify some that are closely tied with Phragmites invasion and removal success and how each influences the microbial community. At each of our sites, we collected the following data on important physical-chemical properties of soil that have a huge impact on plants, soil microbes, and nutrient cycling. Each of these factors determine which species can grow where and which chemical and biological processes can and will take place. Many plant and microbial species can only tolerate a relatively narrow range of variables like pH, salinity, and temperature. And the amount and availability of nutrients like nitrogen and phosphorus have a strong effect on primary production and nutrient cycling. To go along with our soil data, we also examined the major players in nutrient cycling, the plants and the microbes themselves. For each site, we recorded the plant species present, as well as which plant appeared to be dominant. Also measured were plant height and stem density, both of which vary greatly with the species of plants present. Soil was frozen in liquid nitrogen on site, so we could run DNA and RNA analysis back at the lab. RNA analysis is difficult even for people experienced with DNA analysis, because RNA breaks down easily and very quickly. So to extract and purify RNA and DNA from soil samples takes about 7 hours for a batch of 10 samples, a laborious and mind-numbing task that needs to be done in a sterile laminar hood, cleaned regularly to destroy RNases. But our students have done a fantastic job, our undergraduate students, and we have extracted most of our samples since we started in October, though we still have more to go. The next steps will be DNA sequencing for species identification and RNA expression analysis to identify the genes that are being used by the microbes in the soil, particularly those related to nitrogen cycling. While these aren't necessarily our target results, I thought it would be interesting to show some maps of nutrient concentrations at our sites. Nitrogen and phosphorus are the main culprits behind nutrient pollution and eutrophication, and they usually only accumulate from human sources like agriculture and wastewater runoff. So it's very interesting to be able to see where these nutrients are found in high amounts, just among our sites. That's all we have, really. I'm sorry that a lot of you probably can't see. Those are our maps. In a similar fashion, we can display some of the plant metrics that we measured at our sites. Average species richness varies greatly between our sites, and a lot of the sites with Phragmites match up well with the sites that have low richness. But some of the sites display the presence of Phragmites and moderately high species richness. For such sites, it's possible that Phragmites invasion is still in progress and hasn't been able to take over completely like it usually does, or that these sites are recovering after removal treatments. Further analysis would be required to make any conclusions. Once again, there is no real evidence of causation here, just some interesting trends that we noticed between treatments, plant height, and water depth. Of course, plant height is shorter for treated sites because Phragmites has been removed and the vegetation is still recovering from treatments applied the previous year. What is interesting is that water depth is lower on average for treated sites, perhaps as a result of the Phragmites dominance that warranted treatment. One nice pattern that we noticed was that plant species richness seems to improve overall based on this figure here. Plant species richness seems to improve overall after Phragmites removal. And species richness isn't necessarily the best metric for ecosystem health, but it shows a marked improvement from the monospecific dominance of Phragmites. Now onto the microbial data. I apologize for the roughness of the rest of the figures. I just got data back on Wednesday and I quickly ran it through a pipeline so that I could have something to put up for you guys to look at today. And these, yes. So these figures show the abundance at class and phylum level between our three categories, native, treated, and untreated Phragmites. And there aren't really any easily identifiable patterns here, but I will be doing a more formal analysis. Alpha and beta diversity are ways we can measure how diverse our communities are and how different they are from each other. When we look at the Shannon-Weiner alpha diversity in our soils, we see no significant difference between our three site categories, which is somewhat surprising considering the relationship between plant diversity and microbial diversity. This non-metric multidimensional scaling, or NMDS ordination plot, is a great way to visually group the communities and look at beta diversity, or how different they are, using Gray-Curtis dissimilarity. Once again, there seems to be more to the story than just our site categories. This type of ordination plot is a great way to identify environmental determinants of our soil microbial community compositions. Because our site categories don't explain much variance, this can help figure out what does explain variance. pH and salinity are well-known drivers in the microbial community with widely documented effects on species present. It is also known that the nutrients present, like phosphorus and nitrogen, can shape microbial communities and the abundance of functional groups related to the use of such nutrients. These plots show us that there is a significant dissimilarity in microbial communities between sites with native vegetation and the other two categories, but not between treated and untreated Phragmites, and that there is significant dissimilarity between soils where Phragmites is present or absent. This here is a functional prediction plot. Functional predictions can be assigned to our soil taxonomies using Fabro-Tax. Functional predictions aren't super accurate for soil microorganisms, especially in the absence of genus classification, which is difficult to attain just because of the sheer diversity of microorganisms found in the soil, but it can still be used to show some interesting patterns until our RNA analysis is complete. By plotting the abundance of taxa present and the functions they may be able to perform based on taxa predictions, it seems that soils with native vegetation outperform both of the other categories, and that treated soils usually lie somewhere between native and untreated Phragmites. Once again, suggesting that treatments might be successful at returning soils to a pre-invasion state. So since I won the award last year, we've come a long way. Back then, Rachel was a new professor at BYU, and I was her only student. Over the last year, we gained 10 new lab members, and they have been a great help with making my research goals happen. We've been able to finish up soil testing, complete most of our DNA and RNA extractions, presented 2 scientific meetings, now 3, and we even have some preliminary molecular data. Thanks to our wonderful students, we've been able to accomplish amazing things in the past year, more than I could have ever done by myself. But this research project is massive and still very much in progress. I still have a lot of work to do, and while I haven't been able to show you any conclusive results, I hope I have left you all with a little more appreciation for the soil microbes. I appreciate them. And I want to thank everybody who funded, informed, collaborated on this project, anybody who's involved even in the slightest way, all of our undergraduates and friends of the Great Salt Lake. And thank you, everybody. I'm okay if nobody wants to ask questions.
Hi, thank you very much. This is a little aside from your research, but since you're knowledgeable about the area in general. I've read papers from Europe, and then I saw a reference to it to the east coast of the U.S., where they thought that expansion of Phragmites was due to eutrophication, excess nutrients. Does any of your data bear on that, or do you have thoughts on the importance of eutrophication for Phragmites?
That's interesting. Yeah, none of my data is really conclusive yet, but when I looked at Phragmites, like in presence of impaired water quality, it does seem like Phragmites does very well with high levels of nutrients. Like maybe that does facilitate growth, but it's hard to, it's kind of hard to tell what's correlation and what's causation, just based on our study. Thank you.
Hi everyone, my name is Keith Hambrecht. I'm the Invasive Species Coordinator for the Division of Forestry, Fire, and State Lands.
All right, we're good to go. Okay, we're going to do a bit of a tag team effort. So Keith introduced himself. I'm Karin, Kettenring, and I'm a professor in the Watershed Sciences Department at Utah State University. And wherever Wayne went, feel free to ask me that question when we're done here, because I have some thoughts on that as well. So we are going to talk about management, research, and restoration today, focusing on these strong collaborations that have been the basis of our work over the last 10 or so years, and particularly Phragmites control and wetland restoration in this new water reality. So I want to lay the context first as we begin here. So we all know that wetlands are a critical part, inseparable part of the Great Salt Lake ecosystem. But the valuable role that wetlands play is under threat due to dewatering, hydrologic extremes, and invasive species. We are working very actively to develop solutions for management and restoration, and I think these solutions are actually going to serve as a model to other people facing similar situations around the world. We are not alone. And so today we're going to really highlight what we see as our recent successes, as well as the ongoing challenges to the management and restoration of Great Salt Lake wetlands. So we'll first turn towards Phragmites management, and you'll hear us really focus on some great collaborations across management agencies and landowners, and some unique partnerships between managers and researchers. So thank you, Madison, for laying the context, the groundwork for our talk today. So we know Phragmites is a problem. It is found throughout much of North America in wetlands and disturbed habitats. We've got bits and pieces of data in terms of how much we have. One survey that my lab did in particular in 2010 showed that we had about 23,000 acres at that time. It's probably grown quite a bit since then, but maybe leveled off with all the amazing management that's been going on over the last 10 or so years. But hundreds of thousands of dollars are being spent each year on control, and it's just one more threat to our wetlands that are in peril. So part of the reason, much of the impacts of Phragmites, the unwanted impacts, have to do with the fact that it is so incredibly tall and productive. I don't know if Chad Cranny is in the audience right now, but if you know Chad, he's maybe 6'6", he's in the back there. You want to stand up, Chad? You can see how tall he is, right? And so this is Chad Cranny a few years ago, and you can see the Phragmites towering above him. Chad is not a small person, right? And you can see how incredibly dense it is. And so from a habitat, so much of these wetlands, the main management goal is for habitat. This is not good habitat when we have monotypic stands of the species. This productivity is part of the problem. Also fire hazard, also potentially a very thirsty plant, largely because it's so productive. So when I started at USU a little over 15 years ago, we had lots of questions about what is it that makes this plant successful, what is its basic ecology, and how can we better manage it. And so one of the first things that my research group did was to actually survey formally wetland managers along Bear Lake, Utah Lake, and Great Salt Lake. And from that survey, we asked them what were their top management concerns. Phragmites was at the top of the list. It was not surprising, but it was good. It was helpful to hear that. But there were lots of questions about what are best management practices. There was new research coming out from East Coast of the U.S., questions about herbicide timing and type. What about mowing or grazing as alternatives to fire? And then interestingly, at that point, many people were not actively trying to restore the habitat after managing Phragmites, many people were not actually seeding or planting. And we were curious about that. So that, in turn, I will now summarize many years of hard work and labor by a number of graduate students, including Brittany, Christine, and Chad, you can see here. I also put this photo up of Chad and Christine because they spoke at the 2014 Friends of Great Salt Lake Issues Forum. But we embarked on some large-scale experiments with great collaborations across state, federal, private, nonprofit landowners. And we were able to use these collaborations to develop best management practices. We also had some questions about how this plant is spreading, and from our research, from genetic research we were able to identify that seed reproduction was really the critical way that this plant was spreading around. So pulling from all these experiments, we were able to develop best management practices and kind of pass the torch back to many of our manager collaborators. And in particular, I would say one of the key parts of this was identifying that key role of seed production and new management or additional management steps that were really important to control that seed reproduction. Is the microphone going in and out? No, okay, it just sounds like it. Okay, so I'm going to pass the torch to Keith.
Okay, so us managers were then able to take all of Karin's research findings and collaborate with her amazing team to put together best practice guides. And these best practice guides are available for all managers. They really outline the best practices for all of the tools in the toolbox that we have as managers. And we wrote these guides to make them best practices that work within all of our management constraints. So they cover the details of every one of these tools, and it's important to note that not one single tool will remove Phragmites. So one of them alone does not work. We have to use an integrated approach using all these tools. And there's often places where we don't have all the tools available to us, such as some places we're not able to graze, other places we don't have water control. So with all of those best practices and a deep understanding of all these tools in our toolbox, we create these treatment protocols. And just, I know you can't read all the details for this treatment protocol, but in essence, we have phase 1, which is Phragmites removal. And we know that takes many years of treatment. And then we have phase 2, which is our maintenance and restoration phase. So we then take those treatment protocols and we apply them on the ground. This map shows an example area in Farmington Bay, WMA. We take all these areas and we break them down into manageable chunks. And these chunks of project areas are really dictated by water control, the ability to control water, and also the hydrology. So then, after we've broken down these project areas, we can lay out a treatment calendar. And because of all the amazing research, we're pretty good at predicting about how long it's going to take to remove Phragmites from a given area. So we plot a course forward, and then we're able to take each one of those treatment blocks and break them down into the treatments that are needed for each season. And this is crucial for us because we're working on such a large scale. So this allows us to have a common language to communicate amongst all managers. And the details of these treatments are really important. And so in order to stay on those details, we need these calendars, and we need to be communicating across boundaries. So where are we applying all of these treatments, these best practices? We've got over 220,000 acres of wetlands that we're doing it. And I think really the important thing to note on this map is forestry, fire, and state lands is everybody's neighbor on the lake bed, and more importantly, we're the downstream neighbor from everyone. So we have to work collaboratively with everyone. Okay, so again, here's the map showing management areas. This is showing just the state managed wetlands, forestry, fire, and state lands, and the DWR, WMA areas. Here's all of our treatment data overlaid on top of it. For forestry, fire, and state lands, we've done treatments on almost 16,000 acres. For DWR, it's about 21,000 acres. So we're working on a scale of about 40,000 acres. You remember Karin mentioned in 2010, there was approximately 23,000 acres of Phragmites. It's probably expanded since then. But we're now working on about the scale of all the Phragmites that's out there. Another thing to note is some areas where you see less treatment, Bear River Bay, and then along the Shorelands Preserve, we have strategic plans underway for those areas. And then I just want to say really briefly, thanks to all of our amazing contractors that are doing a lot of this work, the aerial spraying, the mowing, and ground treatments as well. And all this treatment, as you can see on the map, we have GPS or GIS data for all of it. So I don't have too much time to get into additional project areas, but we are also doing treatments on the entire shoreline of Utah Lake, which is around 11,000 acres. We do that with Utah County and Utah Lake Authority. And then the Jordan River Corridor. This map shows mapping we had done of Phragmites in the Jordan River Corridor. It's the entire corridor. Small acreage, 200 acres. It's very challenging, and we also have 16 cities and 3 counties, so a lot more collaboration and boundaries. And we work with the Jordan River Commission for that. Okay, so we have these best practices, and we're applying them on thousands and thousands of acres of Phragmites. Are they working? So we have really good data since we're treating the entire shoreline of Utah Lake. We can estimate that as of 2022, we've reduced Phragmites by 74%. That number is also likely much better now. We'll get that at the end of the summer, and I think we're probably over 80% reduction. Great Salt Lake is much, much bigger, 220,000 acres of wetlands, 20 times the size. We don't have the data to give an overall estimate, but we have done some one-off intensive monitoring in certain areas. We utilized Brittany Duncan and her monitoring team, part of UDAF's invasive species monitoring effort, and they did a one-off project for us in at our Turpin West project in Farmington Bay. And we have a before and after photo here. Within 5 years, we were able to go from over 90% Phragmites to over 90% desirable vegetation. And we have transects where Brittany collected data showing we maintain Phragmites cover below 10%. And we're still there now. One thing to note is that after picture, it's maybe hard to see, but mostly what you're looking at in that picture is saltgrass, pickleweed, and then some forbs such as rumex. We also have open water and bare ground. This is the diversity that we need for these birds. And so then another example, inside the impoundments of the DWR managed wetlands. Picture on the left, you can see Phragmites invaded. This is in the middle of the treatment process, so you can see the dead Phragmites laying down. This is the same area 5 years later. It's all open water. And then in the background is cattail. And then also, this past year, DWR tracks all their ground treatments. They covered 6,000 acres on the ground this year, and they only found 15% Phragmites covered in those areas. So our treatments are working really well, but looking ahead, we know that water shortages are going to further hinder reductions in Phragmites. So this is from our management matrix. And you can see when water levels are really low, Phragmites dominates. Basically, we have more Phragmites habitat. We're glad that the lake's on the way back up, but water is very helpful. And furthermore, you know from the research and our experience that we need water to effectively treat Phragmites. So without water, we're really fighting with our hand behind our back. And also, drought stress Phragmites is just really, really hard to kill. The other thing is all of our upstream neighbors, if they're unwilling to treat Phragmites, we've got a major problem because, as Karin pointed out, seeds play a big role. And much of our canals and ditches throughout the entire valley look like that in the photo there. Okay, so we're having all this success. We have these best practices. Now we turn to part 2, which is restoring native plant communities. And here, again, we turn to research to guide the way, because as you'll see from Karin's slide, revegetating all these wetlands is quite complicated.
Okay, so what are we talking about when we're talking about revegetation? We're talking about seeding and we're talking about planting. And planting can be plugs like that photo in the upper right or sod mats like on that previous slide. And I would say as a whole, wetland revegetation is underappreciated in wetlands around the world. It's not just a problem here in Great Salt Lake. And it has been underutilized as a restoration technique. And part of this is the assumption globally, again, that with restoring wetlands, if you remove the stressor, you manage the invasive plants, or you restore the hydrology, the plants will naturally come back because the birds will bring them back. Our research has shown, and many other researchers, decades of research shows that that is not actually the case. Plants might come back, but at a much lower diversity and not necessarily the species that are prioritized for habitat, for instance. And this is really a problem. That photo on the left is an area that has been previously managed for Phragmites. It is prime for Phragmites to reinvade, and that's exactly what we don't want to be doing. We don't want to be spraying more herbicide than we need to. We want to restore that habitat. And so we need to be rapidly reestablishing native plant communities to compete against that Phragmites. And then, of course, having the diversity of species to restore the habitat that we all value. So we have, over the last 7 to 8 years, have really been working on developing and refining best practices for seeding and planting. And this is not an easy undertaking, but again, we have been relying on fantastic collaborations with so many land managers to make this happen across many, many students' projects. I won't be able to give credit to all the fantastic students behind all this as well, but a lot of years of blood, sweat, and tears. There's some basic questions that we need to address to seed and plant. And these questions might at first seem quite easy, but actually they are not. But the first question is, what do we want back and why? And so we need to have a diversity of species for many different reasons. The habitat varies in terms of salinity and hydrology, but we're also, as many people pointed out, facing many more hydrologic extremes. And then, of course, habitat from a pollinator perspective and an avian perspective, we need a whole slew of species. And then we also need some species that are going to grow fast and compete well against Phragmites. So we need to figure out how to get that balance to achieve all these many goals that people have. And part of the challenge is that we have limited experience working with most of these native species. So we just need to understand for every single species we're trying to restore, what is the basic biology, the basic reproduction. And we are working very hard on a portfolio of about 35 plus species, mostly emergent and playa wetland species, to understand these various aspects of their ecology. And really what we're trying to avoid is biotic homogenization. Again, this is happening in restorations around the world where we have just a few species that are coming back. We do not want to lose that diversity, because when we lose that plant diversity, we lose everything else that's associated with it. And one of the biggest challenges is that plants that are hard to work with, if they're more particular, those are the ones that are going to be tossed to the side, and instead, from a researcher standpoint, though, that's actually where we want to dig in, no pun intended, and figure out how to work with them. So one of the big questions, or one of the challenges with working with seeds specifically, which is going to be the solution for restoring tens of thousands of acres, is seed dormancy. So this is an important adaptation that plants have that prevents seeds from germinating during unfavorable conditions. But it can also challenge restoration, because when you put seeds out there, you want them to generally germinate right away, because you're putting them out there when the conditions are right, theoretically. So we are breaking dormancy usually to mimic winter conditions by treating seeds with cold, moist conditions, or scarifying seeds to mimic bird digestion or soil abrasion. But how on earth do you do that at large scales? That's one of the challenges when you have 50-pound bags of seeds, and many of those for revegetation. And again, I'm going to summarize research very briefly here. This is work largely done by Ray Robinson, my former lab manager, and I'll just say that we found a lot of variation across the 30-plus species that we have been working with. Some species in the green there don't actually have dormancy. That's great, it makes it easier to work with. In contrast, some of our Cyperaceae species, those are species, some of the bulrushes and sedges that require not just that winter cold treatment, but also an additional scarification treatment. So they're really challenging to work with, but they're also our most important habitat species. Another fundamental question is, how much seed should we put out there? So if restoration budgets are greatly limited, we don't want to be wasting seeds. They're expensive and they're hard to come by. But what do you do? And from, again, many students' projects, Emily Tarsa, Ray Robinson, Lana Feldman, Maddie Hood, we've seen a lot of mortality of seed. And this is, again, something that's found in dryland restoration, grassland restoration as well. 99% of what you put out there is just going to die because of natural plant limitations. So we have found generally that in terms of seeding, that we need to be seeding about 3 to 5 times the standard seeding rates that people have been using, and that is going to be expensive. But at the same time, some of our research has also shown it doesn't matter how much seed you put out there if you have not done a good job taking care of the Phragmites seeds that are in the soil and the seed bank. And so that is where the biggest investment needs to go before you embark on revegetation. Now I'm going to turn it over to Keith again here.
Okay, so just touching very briefly on our current practices, and all of these are based off of what we've learned from Karin's lab thus far. We use a disc plow to plow the wetlands first. This helps us mitigate fluctuating water levels and create microsites for seed germination. Then we follow behind with a broadcast seeder, and we broadcast seeds that have been stratified for the lab's protocols, so we know that they're ready to germinate rapidly. You can see our disking here in the center, and then below we find that we're getting much better germination in the area that's disked, which is on the left, versus not disked on the right. And then with these current techniques, we're mostly using inland saltgrass and alkali bulrush, and those are the 2 species that we feel like we have good methods for, so we're focusing on those for now. Picture on the right shows some of our seeding success, and we're getting rapid germination and quick establishment, but we're also seeing a ton of challenges, especially with the fluctuating water levels. That's been pointed out multiple times. We had the mega drought and then the highest water level, the highest stream flows recorded the following year with mega snowpack, and this site was actually flooded out by that. So with these current methods, we know that we need to build capacity. So right now we're seeding about 2,000 to 3,000 pounds of seed per year, but we're not covering a very large acreage. And then as Karin mentioned, we have to use high seeding rates to see success, so it's very expensive. And then we're only focused on 2 species right now. We really need to increase diversity, but we have to wait for answers from research in order to do that.
So one example of what Keith was just mentioning is actually played out in Karina Hebert's master's project. She's now with Ducks Unlimited. But in terms of looking to the future, one of the things that we're wondering is how much wetland habitat can be restored, and that is really going to be constrained by water availability. So in May of 2021, Karina, with collaborators, went out and seeded her sites. She went back in June, and success, lots of alkali bulrush seedlings. We were seedlings coming up. We were super excited. But then July, August, drought, extreme heat, and then followed by late summer rains. And so when she went back and surveyed later in the year, basically all of those alkali bulrush seedlings had died, and the Phragmites had rebounded because of those summer rains. So that really underscores the challenges that we're facing. And we wanted to get a sense of what are those challenges more broadly. So one of the things that we have done is actually surveyed wetland managers throughout the Intermountain West and asked them about their revegetation practices in case there was, there's lots of things we can learn from people outside of the Great Salt Lake watershed. And then what are the challenges that they face? And so they identified both environmental challenges, which you can see on the left-hand side there, the primary environmental challenges that managers are facing, drought and climate change, invasive plant, sorry, I can't actually read my slides here, invasive plant species, and timing and delivery of water. Those are their biggest challenges for revegetation specifically. And then with respect to capacity-related challenges, their biggest challenges relate here to budgets, not enough personnel, and the logistics of trying to seed and plant large acres. So the question I think that we need to address is what costs are we willing to incur to have these higher seeding rates? We potentially need to be reseeding sites when we have high mortality. And are there resources available that are proportional to the scale of the need here for restoration? And similarly, we wanted to know what seeds and plants are out there. Can we actually get what we need for revegetation? And so we just wrapped up a survey of native plant vendors. And one of the things that we asked them was, what are the offerings that you have? And we found on average, vendors had less than 4 aquatic species, less than 15 emergent and playa species that they were selling. And these are most likely what we call workhorse species that we know will do well, like saltgrass and alkali bulrush. This is a problem when we're again talking about diversity. So if you look at the larger pool of species that we might want to be restoring, we have about 1,500 species in wetlands in the Intermountain West. Not all of them are going to be along Great Salt Lake, but there are a lot of species out there, but they're not available for purchase. And so access to diverse native wetland seeds are really going to be a big challenge. So we're wrapping up here, but again, I just want you to know that we're part of a much broader global effort to restore ecosystems through seed-based restoration, and the wetland work that we are all collectively doing in this region are actually leading the way. And one great example of this is a video or a documentary that was released last year. You can see the URL on the right-hand side there, and it's a documentary on native seeds supplying restoration, all about seed-based wetland restoration. You can see, though, in blue there are 3 locations in Utah. They came to Utah to see what Division of Forestry, Fire, State Lands, Division of Wildlife Resources, what people were doing, and you can see some of Farmington Bay Waterfowl Management Area was one of their prime filming locations. Super cool documentary, so you're going to want to check it out.
Okay, so in closing, we just want to say we remain optimistic about the future of Great Salt Lake wetlands. However, we emphasize the vital importance of securing sufficient water, obtaining enough funding, having a diversity of species, and supporting strong collaboration. On a few of these items, I want to say we did get some excellent support from the state legislature this past session for funding for forestry, fire, and state lands. So we're grateful for that, and hopefully it keeps up. And then on the collaboration note, I just want to say thanks to all of the many collaborators for all this work. There's a ton. Specifically, Utah County Weed Control Department has done an excellent job at Utah Lake, and then all of the DWR, WMA managers on Great Salt Lake, and specifically my counterpart, Chad Cranny, for his work with DWR. And then I also really want to thank Karin and the Wetland Ecology and Restoration Lab for all their amazing work.
Also, we have free aquatic plant guides. We didn't talk about this at all, but we have about 20 more of these if anybody would like one. Talk to Sam Kurkowski.
I guess not.